Yes. Good morning, dear students. So today we are going to discuss with another important biomolecule, that is nucleic acid. So this nucleic acid, wherever you come across with the word nucleic acid, you have to remember the two terminologies. One is DNA and the another one is RNA. If you hear this word nucleic acid, the nucleic acid means it consists of two important components. One is either it may be DNA or it may be RNA. So what this nucleic acid? So the nucleic acid is a content or a biomolecule which is very important, which carries a heredity information or you can also say it as a which carries a, or you can also term it as genetic material, which is nothing but DNA or RNA. So this nucleic acid was first coined by a person called as studied by a person called as Frederick Mischer, and later the term nucleic acid was coined by, or the name was given by Altman. The name was given, or the name was coined by a person called as Altman. Next, now the nucleic acid, what is this nucleic acid? What is a basic, or what is a building block, or what is a unit of nucleic acid? What is this nucleic acid made up of? As we have discussed, the protein, it is made up of amino acid, the carbohydrate, it is made up of sugar units, lipids, it is made up of fatty acid and glycerol. In the same way, the nucleic acid is made up of nucleotides. The nucleic acid is made up of nucleotide. Hence, the nucleotides are the building blocks of nucleic acid. Again ಹೇಳ್ಬೋದು the nucleic acid ಅಂದ್ರೆ ನಾವಾಗಲೇ ಯಾವ್ದು ಹೇಳಿದೆ, DNA and RNA. So hence this DNA and RNA, both these two are made up of nucleotides. What it is made up of? It is made up of nucleotides. Now, what does this nucleotide consist of? The nucleotide, it consists of three major elements or three major components. The nucleotide, it is consist of three major elements. One is pentose sugar, nitrogen bases, and the third one is phosphate group, or it is also called as phosphoric acid. Phosphate group, or it is also called as phosphoric acid. It is abbreviated, or it is given in the formula H3PO4. H3PO4. Now we'll study one by one. What is this pentose sugar, what are nitrogen bases, what is phosphate group? See, this is a very basic concept which you have already studied in your lower classes also, but by taking this basic concept in detail, we are going to study about the structure of the DNA. Understood? So first we'll take up with the basic concept. So it is made up of three units. The nucleotide, it is made up of three units: pentose sugar, nitrogen bases and phosphate group. So first I have taken with the pentose sugar. In the pentose sugar, it is of two types. One is deoxyribose and another one is just ribose. Deoxyribose, and another one is ribose. As the name itself, pentose sugar, it has 5 carbon atom, H5, C5, H10, O5, whereas here C5, H10, O4. Deoxy, there is one oxygen less, whereas here there is one oxygen. So C5, H10, O5 is the molecular formula of ribose sugar, deoxyribose sugar, as well as ribose sugar. So now in this 2 sugar in it, the DNA is made up of deoxyribose. The DNA, it stands for deoxyribonucleic acid. RNA, it stands for ribonucleic acid. So the ribose sugar, it is seen in RNA, whereas the deoxyribose sugar, it is seen in DNA. So the DNA and RNA, both it contains pentose sugar, it contains nitrogen bases, as well as it also contains the phosphate group. Whereas what is the difference between now DNA and RNA? The first difference is, in the DNA there is a pentose sugar. That pentose sugar is deoxyribose sugar, where in the DNA it is made up of deoxyribose sugar, whereas in RNA it is made up of ribose sugar. So there is, now we have got one difference. What is the difference between DNA and RNA is, it differs in pentose sugar. What is the pentose sugar? Deoxyribose sugar that is seen in DNA, whereas in RNA it consists of ribose sugar. So this is one of the main basic concept. Next is nitrogen bases. Next one is nitrogen bases. So in the nitrogen bases, it is again classified into 2 categories based on the ring structure. One is purines and the another one is pyrimidines. Nitrogen bases ಅಲ್ಲಿ 2 ತರ ಇದೆ, ಒಂದು purines ಇನ್ನೊಂದು pyrimidines. So wherever you come across with the word nitrogen bases, you have to remember this 3 alphabet A, T, G, C. It is in case of DNA, whereas in case of RNA it is AUGC. Instead of T there will be a uracil. So nitrogen bases ATGC, adenine, guanine, cytosine and thymine, whereas there adenine, uracil, guanine and cytosine. So now this 4 nitrogen bases, they are classified into 2 categories based on their ring system. The purines, it is made up of double ring structure, whereas pyrimidine, it is made up of single ring structure. Purines, it is made up of double ring structure, whereas pyrimidine, it is made up of single ring structure. Now in purines there are 2 types. One is adenine, another one is guanine. AG comes under the purines, whereas pyrimidines AT AG ಇಲ್ಲಿ ಹೋಯ್ತು ಅಂದರೆ ಇನ್ನು ಉಳ್ಕೊಂಡಿರೋದು ಯಾವುದು? pyrimidines ಅಲ್ಲಿ ಥೈಮಿನ್, ಸೈಟೋಸಿನ್, ಇಲ್ಲಿ ಇನ್ನೊಂದು ಉಳಿತಲ್ಲ ಯಾವುದು? ಯುರಾಸಿಲ್. But this uracil, it is seen in RNA, whereas the thymine, it is seen in DNA. ಅಷ್ಟೇ ಡಿಫ್ರೆನ್ಸ್. ಥೈಮಿನ್ ಎಲ್ಲಿ ನೋಡ್ತೀವಿ? ಟಿ ಬರೋದು ಯಾವಾಗಲೂ ಡಿ ಎನ್ ಎಲ್ಲಿ ಮಾತ್ರ. ಯು ಅನ್ನೋದು ಬರೋದೇ ಇಲ್ಲ ಡಿ ಎನ್ ಎಲ್ಲಿ. ಇನ್ಸ್ಟೆಡ್ ಆಫ್ ಟಿ ಇನ್ the RNA, uracil is seen. So there are totally 4 nitrogen bases, A, T, G, C, or in RNA instead of T it is replaced with U, uracil. So what is this nitrogen bases? The nitrogen bases are the content which is mainly classified based on their structure, ring structure. The purines, it is made up of double ring structure. ಈ ಥರ ಡಬಲ್ ರಿಂಗ್ ಸ್ಟ್ರಕ್ಚರ್ ಇರುತ್ತೆ. ಯಾವುದು? ಪ್ಯೂರಿನ್ಸ್. Whereas pyrimidines, it is made up of single ring structure. The purines, it is made up of adenine, guanine, AG. Alphabet ನೆನ್ಪಿಟ್ಕೊಳ್ಳಿ. AG means purine. Whereas thymine, cytosine and uracil. If you remember thymine, automatically you can write uracil. But the uracil, it is only present in RNA. Whereas thymine, it is present only in DNA. So the thymine cannot be seen in RNA. In the same way, the uracil cannot be seen in DNA. But both it belongs to which one category? Pyrimidines, thymine and cytosine. Now, so this is about the nitrogen bases. So pentose sugar, there are 2 types of pentose sugar. One is deoxyribose pentose sugar and another one is ribose pentose sugar. So this deoxyribose sugar, it is seen only in DNA, whereas in RNA there is a ribose sugar. Next, coming to the nitrogen bases. The nitrogen bases, it consists of 4 content. One is adenine, guanine, cytosine and thymine. If it is written, if I am using the word thymine, it is understood it comes in a DNA or it belongs to DNA. Not comes, it belongs to DNA if I am using the thymine. If I am using adenine, uracil, guanine and cytosine, if I use the word uracil, you have to be remember it is related to RNA. There is no DNA there. Next, the nitrogen bases, it is classified into 2 categories. One is purines and pyrimidines. On what basis it is classified? And it is based on their ring structure. If it is made up of 2 double ring structure, then it is purines. If it is made up of only single ring structure, then it is pyrimidines. Next, the purines, it is made, the best example for the purines is one is adenine, another one is guanine. Whereas in case of pyrimidines, it is made up of thymine, cytosine and uracil. Thymine, cytosine and uracil. So this is about the nitrogen bases. Next, there is another one important thing what you have to remember in the nitrogen bases, that is in the nitrogen bases, the purines always pairs with pyrimidine. The purines always pairs with the pyrimidines. So purines ಯಾವ್ಯಾವುದು? adenine ಮತ್ತು guanine. pyrimids ಯಾವ್ಯಾವುದು? thymine, cytosine ಮತ್ತು uracil. Purines always has to pair with pyrimidine. ಅರ್ಥ ಆಯ್ತಾ? The purine always has to pair with the pyrimidine. Now what pairs with what ಅಂದರೆ adenine always pairs with thymine with 2 hydrogen bond. 2 hydrogen bond. ಯಾವುದು? The adenine always pairs with thymine with 2 hydrogen bond. I mean in between adenine and thymine we can see double bond of which one? Hydrogen. Whereas the guanine and cytosine are always pair and we can see 3 hydrogen bond between the guanine and cytosine. Hence there is one rule that the purines, the number of purines are always equal to the number of pyrimidines. The number of purines is always equal to the number of pyrimidines. This rule number of purines is always equal to the number of pyrimidines, that is A plus G is equal to T plus C. This rule is called as Chargaffs rule. Very important. This was first studied by a person called as Chargaff, hence the name called as Chargaffs rule. So what is according to this rule antandre? In this rule they say the purines always pairs with pyrimidine. Yes, the purine always pair with pyrimidine. So hence the adenine will always pairs with thymine. It will not pair with cytosine. The adenine will always pairs with thymine with a 2 hydrogen bond in between. Whereas the guanine always pairs with the cytosine with a 3 bond. With a 3 triple bond, that is hydrogen bond itself, both contains hydrogen bond, but the 2 hydrogen bond is seen between adenine and adenine and thymine, whereas the 3 hydrogen bond it is seen between guanine and cytosine. Hence the person called as Chargaff, he put one rule, or he put forwarded one rule called as the number of purines is always equal to the number of pyrimidines. A plus G is always equal to T plus C. This rule is called as Chargaffs rule. So this is about the nitrogen bases. Now next, now the next content is phosphoric acid, that is H3 PU-4 minus, or general we can say it as a phosphate group so the phosphate group and the pentose sugar ನಾವು ಹಿಂದೆ ಓದಿರಿ ಪೆಂಟೋಸ್ ಶುಗರ್ ಯಾವ್ಯಾವುದು, ಒಂದು ರೈಬೋಸು, ಇನ್ನೊಂದು ಡಿಆಕ್ಸಿರೈಬೋಸ್. ಲೆಟ್ ಅಸ್ ಟೇಕ್ ನಾವು ಡಿ ಎನ್ ಎಲ್ಲಿರುವಂಥ ಪೆಂಟೋಸ್ ಶುಗರ್ ಯಾವುದು, ಡಿಆಕ್ಸಿ ರೈಬೋಸ್ ಪೆಂಟೋಸ್ ಶುಗರ್. ಈ ಡಿಆಕ್ಸಿ ರೈಬೋಸ್ ಪೆಂಟೋಸ್ ಶುಗರಿನ ಕಾರ್ಬನ್ ಆಟಮು ಮತ್ತು ಇಲ್ಲಿರುವಂಥ ಹೆಚ್ಚು ಇಲ್ಲಿ ಎಸ್ಟರಿಫಿಕೇಶನ್ ಆಗುತ್ತೆ. ಹೆನ್ಸ್ ಇಟ್ ರಿಸಲ್ಟ್ಸ್ ಇನ್ ಅ ಫಾರ್ಮೇಷನ್ ಆಫ್ ಅ ಬಾಂಡ್ ಕಾಲ್ಡ್ ಆಸ್ ಫಾಸ್ಫೋ diester bond. So just remember, there is a bond formation occurs between the phosphate group and the pentose sugar, and that bond is called as phosphodiester bond. What is phosphodiester bond means? It is a bond which occurs between the phosphate or phosphoric acid with the pentose sugar. Either the pentose sugar may be ribose or it may be a deoxyribose. That result in a formation of a bond called as phosphodiester bond. See this phosphodiester bond, it is not formed a bond between the nitrogen bases. Nitrogen bases madhya alla. Nitrogen bases purine ಮತ್ತೆ pyrimidine in ಮಧ್ಯೆ ಇರೋ ಬಾಂಡ್ ಯಾವುದು ಅಂದರೆ ಹೈಡ್ರೋಜನ್ ಬಾಂಡ್. A ಮತ್ತು T in ಮಧ್ಯೆ ಇರೋದು ಇದು ಬಾಂಡ್ ಹೈಡ್ರೋಜನ್ ಬಾಂಡ್ ಅದು. Whereas the phosphodiester bond, it is seen between phosphate group and the pentose sugar. Now there is another one word called as nucleoside. Now ಹಿಂದೆ ಹೇಳಿದ್ದೆ ಅದು, the structural and functional unit of this nucleic acid is called as nucleotide. ಅಲ್ಲಿ ಏನಂತ ಹೇಳಿದ್ದೆ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್. ಈಗ ಅದೇ ಥರ ಇನ್ನೊಂದು ಸಿಮಿಲರ್ ವರ್ಡ್ ಇದೆ, ಅದನ್ನು ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ ಅಂತ ಅಂತೀವಿ. ದೆನ್ ವಾಟ್ ಈಸ್ ದ ಡಿಫ್ರೆನ್ಸ್ ಬಿಟ್ವೀನ್ ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ ಆ್ಯಂಡ್ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಅಂತಂದರೆ, ದ ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ ಇದು ಯಾವ್ಯಾವುದರಿಂದ ಮೇಡಪ್ ಆಗಿತ್ತು, 3 ಯೂನಿಟ್ಸ್. ಒನ್ ಇಸ್ ಪೆಂಟೋಸ್ ಶುಗರ್, ನೆಕ್ಸ್ಟ್ ಒನ್ ಇಸ್ ನೈಟ್ರೋಜನ್ ಬೇಸಿಸ್, ಆ್ಯಂಡ್ ದ ಥರ್ಡ್ ಒನ್ ಇಸ್ ಫಾಸ್ಫೇಟ್ ಗ್ರೂಪ್. H3PO4 minus phosphate group or phosphoric acid H3 ಅಂತ ಬರ್ತಾರೆ phosphoric acid. So ಇದು ಮೂರು ಸೇರಿದ್ರೆ ನಾವೇನಂತೀವಿ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್. Then what is this nucleoside ಅಂತಂದ್ರೆ the nucleoside it consists of pentose sugar plus nitrogen bases. It consists of pentose sugar. Pentose sugar ಅಲ್ಲಿ ಮತ್ತೆರಡು, either it may be deoxy or it may be ribose. ಒಂದು ಡಿಯಾಕ್ಸಿ ರೈಬೋಸ್ ಇರ್ಬೋದು ಅಥವಾ ಜಸ್ಟ್ ರೈಬೋಸ್ ಇರ್ಬೋದು, ಯಾವ್ದಾದ್ರು ಇರ್ಬೋದು. ಇನ್ ಕೇಸ್ ಆಫ್ RNA ತೊಗೊಂಡ್ರೆ RNAದು ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ ಬರಿಬೇಕು ಅಂದರೆ ರೈಬೋಸ್ ಶುಗರ್ ಬರುತ್ತೆ. DNA ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ ಬರಿಬೇಕು ಅಂದರೆ ಡಿಯಾಕ್ಸಿ ಬರುತ್ತೆ, ಪೆಂಟೋಸ್ ಶುಗರ್. So don't get confused. If you write down it in a proper systematic way, it will be very easy. Don't get confused. So the pentose sugar, it may be either deoxy or it may be ribose. Along with that, it consists of a nitrogen bases. Nitrogen bases, the same thing, ATGC purines. In case of you take ATGC in case of DNA, AUGC in case of RNA. So no change anything in the nitrogen base. The only thing which lacks in nucleoside is phosphate group. So this phosphate group is completely absent in case of nucleoside, but it is present in nucleotide. If the phosphate group is not there, then it is considered as nucleoside. If the phosphate group is there, then between pentose sugar and nitrogen base, then it is called pentose sugar and phosphate group. If they together they form the phosphodiester bond. So if the pentose sugar, if the H3PO4, that is phosphoric acid, is absent, it is considered as nucleoside. For this nucleoside, if I add phosphate group, then the nucleoside plus phosphate group together it forms nucleotide. So hence the nucleotide, it is a combination of nucleoside plus phosphate group. So the nucleoside plus phosphate group together it forms nucleotide. Then what is lacking in nucleoside antandre, that is a phosphate group. The rest thing it remains same, there is no change. So this is what the general discussion of DNA and RNA. Now the DNA, the adenine, guanine, cytosine and thymine, they are all the nitrogen bases. Now in DNA, how along with the nucleoside and nucleotide, how it is called as deoxyadenilic acid, ಡಿಯಾಕ್ಸಿಅಡೆನೈನ್ ಕಂಪ್ಲೀಟ್ ಅದನ್ನು ಹೇಳೋದಾದರೆ ಹೆಂಗೆ ಅಂದರೆ ಡಿಯಾಕ್ಸಿಅಡೆನಿಲಿಕ್ ಆ್ಯಸಿಡ್, ಗ್ವಾನೋಸೈನ್ ಆ್ಯಸಿಡ್, ಆ ಥರ ನಾವು ಅದನ್ನು ಆ್ಯಸಿಡ್ ಅಂತ ನೇಮ್ ಮಾಡಿ ಹೇಳ್ತೀವಿ. ಯಾಕೆ ಆ್ಯಸಿಡ್ ಅಂದರೆ ಬಿಕಾಸ್ ವಿ ಆರ್ ಆ್ಯಡಿಂಗ್ H3PO4, ದಟ್ ಇಸ್ ಫಾಸ್ಫೋರಿಕ್ ಆ್ಯಸಿಡ್, ಡಿಯಾಕ್ಸಿಗ್ವಾನಲಿಕ್ ಆ್ಯಸಿಡ್, ಡಿಯಾಕ್ಸಿಸೈಟಿಡಿನಿಕ್ ಆ್ಯಸಿಡ್, ಡಿಯಾಕ್ಸಿಥೈಮಿಡಿನಿಕ್ ಆ್ಯಸಿಡ್ ಅಂತ ಆ್ಯಸಿಡ್ ಅಂತ ಲೇಬಲ್ ಮಾಡಿ ಹೋಗ್ತೀವಿ. ಅದೇ ನೀವು RNAಗೆ ತೊಗೊಂಡ್ರೆ ಯು ಕ್ಯಾನ್ ಸೇ ಇಟ್ ಆಸ್ ರೈಬೋನ್ಯೂಕ್ಲಿಕ್ ಆ್ಯಸಿಡ್, ಅಂಡರ್ಸ್ಟುಡ್, ಗ್ವಾನೋಸೈನ್ ಮೋನೋಫಾಸ್ಫೇಟ್, ಅಡಿನೋಸೈನ್ ಮೋನೋಫಾಸ್ಫೇಟ್ ಅಂತ RNAಯಲ್ಲಿ ಬರ್ತಾ ಹೋಗುತ್ತೆ. ಸೊ ದಟ್ ಇನ್ ಡೀಟೇಲ್ ವಿಲ್ ಸ್ಟಡಿ. ಸೊ ದಿಸ್ ಇಸ್ ಅಬೌಟ್ ದ ನ್ಯೂಕ್ಲಿಕ್ ಆ್ಯಸಿಡ್. So just I have given a brief introduction towards the biomolecule. So first we have studied with the carbohydrate, the sugar units, basic. Based on the sugar unit, the carbohydrate, it is classified into 3 categories that we have discussed. Next is we have discussed with the proteins. So proteins are another type of building blocks which is made up of amino acid. So there are 20 amino acids that we have discussed regarding the general brief introduction about the proteins. Next we have discussed with the fat. Fat is made up of 2 contents, that is lipids is made up of 2 contents, in the lipid. One is fatty acid, it is made up of fatty acid, esterification of fatty acid, and glycerol itself, it forms the lipids. So lipids alli matte it is of different type, simple lipids, conjugated lipids, derived lipids, ಮತ್ತು ಅದರ ಸ್ಟಿರಾಯ್ಡ್ಸ್ ಅನ್ನೋದೊಂದು ಕಾನ್ಸೆಪ್ಟ್ ಬರುತ್ತೆ. ಅದಾದಮೇಲೆ we have discussed with the enzymes. So enzymes, they are the biocatalyst which alters the rate of reaction or which enhances the rate of reaction, understood. And this enzyme itself, it will not undergo any change. It will, after completion of the reaction, it will take up another substrate and it will continue with the reaction. We have discussed regarding the enzymes. So all enzymes are proteins, but all proteins are not enzymes. So that is very important word. Next, after the enzymes, we have come across with the concept of nucleic acid. So next, in the future classes of this nucleic acid, you are going to study with the structure of DNA, types of RNA, everything we are going to discuss there. So just I have given an introduction. So in tomorrow's class, just I am going to give an introduction towards the morphology of the flowering plants. That is, if we take a plant, what are the morphological characters we can see in the plant, as well as we'll study with the different types of tissues. In the upcoming classes, we are going to study with that concept. Thank you.